I'm Dr. 𝗛𝗦𝗬𝗢𝗚, hematopathologist. I'm going to be with you during these two lectures discussing an important base for the next year, inshallah, in the third stage, talking about the bone marrow histology and the process of haemopoiesis. Starting with the bone marrow. What is the bone marrow? The bone marrow is a tissue that occupies the cavities under the cortex within the honeycomb of trabecular bone. So it is a tissue that occupies a special space within the bones. This bone marrow is either red or yellow. The red bone marrow is the part of the bone marrow that contains the haemopoietic tissue. And here, the process of haemopoiesis takes place. While the yellow bone marrow is actually a adipose tissue present with the red bone marrow. It's not responsible for any synthesis of any haemopoietic cells. This is illustrated in the following picture, showing the red bone marrow here and the yellow bone marrow here. In children, most of the bones contain haemopoietic bone marrow, means that all the bones in children contain a red bone marrow. In contrast to the adult, where the red bone marrow is only present in special sites, including the skull, the sternum, the scapulae, vertebrae, ribs, pelvic bone, and the proximal ends of long bone. This picture will show the distribution of the bone marrow, actually the red bone marrow all over the body in children, and the limited spaces or the limited sites for bone marrow in adults. The bone marrow is composed from parenchyma and stroma. The parenchyma contain the haemopoietic cells in different stages of maturation, starting from immature cells progressively into more mature one. And also the parenchyma contain an important cell, which is the haemopoietic stem cell. While the stroma is composed from fat cells, histiocytes, fibroblasts, blood vessels, and intracellular matrix. This picture illustrates the constituents of the stroma, including the macrophage, the endothelial cells. These are the fibroblasts under the endothelial cells, fat cells, extracellular matrix, giving the shape or the architecture of the bone marrow in order to an appropriate and well-controlled haemopoietic process takes place. The haemopoietic precursors or the haemopoietic cells receives their nutrient, vitamins, hormone, regulatory factors through this extracellular matrix, which also contribute to the regulation of the cell cycle and cell differentiation. The blood supply for these haemopoietic cells and to the bone marrow consists of two systems, either from the periosteal arteries, which located under the periosteum of the bone, which gives off branches to the bone marrow after they penetrate the bones, and the blood drain from the blood cavity is through the central vein. The microvasculature of the bone marrow comprises a network of sinusoids. Haemopoiesis only occur in the interstitial space between these sinusoids. الساينوسويدز مهمة in order to ensure that a good nutrient will be received to these haemopoietic precursors. Normal bone marrow also contain a network of fine branching reticulin fibers. These are synthesized by these fibroblasts. The reticulin fibers will pass between the parenchymal cells and provide the extracellular matrix for the bone marrow. There is higher concentration of thicker fibers around the arterioles and near the endosteum. The bone marrow structure, the parenchyma of the bone marrow is composed from the hemopoietic cells, including the precursors and mature cells of erythroid. These are the erythroid cells, the myeloid cells, and the megakaryocytes. Megakaryocytes is number 10, 11, 12, 13, 11 and 12 are precursors of granulocytes, and normoblast, erythrocyte in number 1 and 2. These are the component of the hemopoietic element. This is a very nice picture, illustrate each part of the bone marrow structure. Please use it when you read the PDF during studying. The second part of our lecture will describe the process of blood formation, how blood formed. Actually, the hemopoiesis process includes three separated processes: the erythropoiesis, the myelopoiesis, and the thrombopoiesis. The erythropoiesis is the process of red blood cell formation. The myelopoiesis is the process of white blood cells or granulocyte formation. And the thrombopoiesis, it is the process of platelet formation. The entire process is under the control of glycoprotein hormone called the hemopoietic growth factors. For example, granulocyte colony stimulating factor. The hemopoiesis takes place in different sites. In adult, we say the size of the bone marrow is limited to specific sites such as the vertebrae, the ribs, the sternum, the skull, and here the process of hemopoiesis takes place. While in infants, the bone marrow is found in children in general, found all bones, so the process of hemopoiesis could take place in all over the body. In fetus, the process is different. The site of hemopoiesis are going to be changed according to the gestational stage. During the first trimester or during the first 3 months of a pregnancy, the process of hemopoiesis will take place in the yolk sac. In the second trimester, with more development of the liver and spleen, they are going to be the size of hemopoiesis. While during the end of pregnancy, near to the end, the bone marrow will take place as a site of hemopoiesis. This is the normal architecture of the bone marrow. The whitish vacuoles here refer to the adipose tissue, the yellow bone marrow, while the red colored cells or pinkish in color are the hemopoietic cells, the parenchymal part of the bone marrow. In order to start the process of poiesis, a single cell have a major role. This cell have a special characteristic. The first one, that it has an ability for self-renewal, and it gives rise to separate or to all cell lineage. So it is an important cell in order to keep the hemopoietic environment in a steady state by the presence of this hemopoietic stem cell. The concentration of this cell is about 1 per 20 million nucleated cell in the bone marrow. So it has very small concentration, and it has the appearance of a small or medium-sized cell, looks like a lymphocyte. From this hemopoietic stem cells, different cell lines arise. The main two cell lines are the common lymphoid progenitor cell, and the second line is the common colony forming unit, granulocyte, erythroid, monocyte, and megakaryocyte. And from each of these two main groups, differentiation takes place. From the common lymphoid progenitor cell, we can get the B and T lymphocyte with the natural killer cell. And from the colony-forming unit, granulocyte, erythroid, macrophage, megakaryocyte, different main progenitors are produced. For example, the colony-forming unit granulocyte, basophil, eosinophil, granulocyte, monocyte progenerator, megakaryocyte progenitor, and the erythroid progenitor. The end results are different cells, but actually all of them arise from this pluripotent stem cell. This pluripotent stem cell pluripotent stem cell have the shape of a small to moderate size cell which looks like a mature lymphocyte. This table will summarize the components of the blood, their characteristic feature, and the lifespan they spend in the peripheral blood, the number or the concentration of each of these cells with their function. And actually, we are going to describe each of them separately in the next few slides. Starting with the red blood cells. The red blood cells are synthesized through a process called the erythropoiesis, which is a highly complex and regulated process that makes about 10 to the power 12 new erythrocytes each day. The lifespan of the red blood cells is about 120 days. Erythropoiesis will start from stem cell, from actually the haemopoietic stem cell, passing through the colony-forming unit granulocyte, erythroid, monocyte, and megakaryocyte to what is called the burst-forming unit erythroid. The burst-forming unit erythroid will give erythroid colony-forming unit. This erythroid colony-forming unit will differentiate or will be differentiate to the first recognizable erythrocyte precursor in the bone marrow called the pronormoblast. So the pronormoblast is the first cell that we can recognize in the bone marrow with a special characteristic feature. This pronormoblast will give rise, to series of progressively smaller normoblasts will be divided to smaller cells called the normoblast. The nucleus is finally extruded. We will get rid of the nucleus. As we know, the red blood cells is a nuclear cell from the normoblast within the bone marrow, and a reticulocyte stage will result. The reticulocyte stage is a stage before full maturation of red blood cells. It is a cell that contains ribosomal RNA. It contains remnant actually of this ribosomal RNA, and it's still able to be synthesized hemoglobin. It spans only 1 to 2 days in the marrow and circulate also in the peripheral blood for 1 to 2 days. Actually, the reticulocyte, the importance of the reticulocyte is to assessing the function or the activity of the bone marrow. It can give us a reflect of how much our bone marrow is active. When RNA is completely lost from this reticulocyte, a completely pink staining mature erythrocyte will result, which is non-nucleated biconcave disc. The red blood cells, the entire process of the red blood cell synthesis, the erythropoiesis, is under the control of an important protein that is 90% of it synthesized from the kidneys or 10% synthesized from the liver, called the erythropoietin. The erythropoietin has no preformed stores. Actually, when the body needs more RBC to be synthesized in order to transfer oxygen or in order to perform its function, will cause activation of these interstitial cells of the kidney to synthesize more erythropoietin. So the stimulation of the production of this hormone is under the control of oxygen tension in tissue of the kidneys. The red blood cells contain the hemoglobin, which is specialized protein responsible for the process of gas exchange to carry the oxygen from the tissues and return back the carbon dioxide from the tissues to the lung. Each molecule of Each molecule of erythropoietin, this is the erythropoietin that controls the process of erythropoiesis. Each molecule of normal, hemoglobin contain hemoglobin A, that is called the adult hemoglobin, and constitute up to 96 to 98% of the total adult hemoglobin. Composed of the chemical structure of this adult hemoglobin is two chains called the alpha chains and two beta chains. So a pair of alpha chain, polypeptide chain, and a pair of beta chain. The hemoglobin F, has the minor constituent. It's actually the major hemoglobin during the stages of embryogenesis, while in adult there will be during delivery, after delivery, there will be switch of this fetal hemoglobin to mature adult hemoglobin. In adult, the constituent or the remnant of the fetal hemoglobin is only 0.5 to 0.8%. The fetal hemoglobin composed of a pair of alpha and gamma chain. The last hemoglobin present in our blood as adults is called the hemoglobin A2, and actually these are minor quantities in our blood in percentage of 1.5 to 3.2 or 3.3, composed of pair of alpha and delta chains. وياي طلاب؟ هسه منو الميجر components of adult hemoglobin inside our bodies, and what is the major hemoglobin in fetus? شنو هو الميجر hemoglobin بالفيتس؟
hemoglobin F.
Excellent. One adult?
Hemoglobin A.
Excellent. So can you think why there is change, or why this switch takes place from embryonic period to adult period or post delivery period? شنو الفرق بين hemoglobin F و A? Can you search for this? This is a homework for you to look for the cause of switching of hemoglobin from fetus hemoglobin to adult hemoglobin after delivery. Why this switch takes place? This is a homework for you. You have to send this the answer of this question, please. The end result is a flexible biconcave disc. A flexible, very important to know that the RBC have this characteristic feature, the flexibility that allow the red blood cells to pass through small microvasculature without destruction. This flexibility allow the red blood cells to pass through the small vasculature without destruction. The size of the red blood cells is about 8 micrometer in diameter, and as we said, it have a lifespan of about 120 days. According to this, what is the main function of the red blood cell? What is the main function of the red blood cell? ها طلاب هسه ورا ما عرفنا exchange gases through
hemoglobin pigment.
Excellent. Now we are going to shift to the other process from haemopoiesis, which is the granulopoiesis. The white blood cells are called the granulocytes. Actually, they are called the granulocytes or the leukocytes. Leuko means white, cytes mean cells. So they are called also leukocytes. The process, the white blood cells, may be divided actually into two main groups, the phagocytes and the lymphocytes. The phagocytes have the ability for what? ليه سميناهم phagocytes?
Phagocytosis. They can engulf bacteria.
Excellent, because they have the ability for engulfment of foreign bodies. So they are called phagocytes. The phagocytes actually compromise the innate immune system. innate immune system اللي هو الموجود عندنا اللي هو does not need activation. So they have very rapid or very quick reaction after an infection. بينما lymphocytes, they are responsible for the adaptive immune response, which needs an immunological stimulation or immunological memory to be activated. Example on this is the vaccination process. Phagocytes also can be divided into two main groups, the granulocytes and the monocytes. So the white blood cells are divided into phagocytes and lymphocytes. Phagocytes responsible for which part of the immune system? For the innate immune system, while the lymphocytes responsible for the adaptive immune system. The phagocytes also again are divided into monocytes and granulocytes. The monocytes and the granulocytes. The granulocytes including the neutrophils, the eosinophils, monocytes, basophils and lymphocytes. طلاب ما بطلبها من عندكم هاي الأرقام. These are not actually only to be as an examples, the different concentration to show that there is different concentration of the phagocyte component, but actually you does not have to know them. Okay? حتى لا تحتارون بعدين. These are the different granulocytes, starting with the first picture showing the neutrophil. The neutrophil have a special characteristic appearance, which is a dense nucleus. See, the nucleus consists of, this nucleus actually is lobulated, composed of 2 to 5 lobes. This is 1, 2, 3, 4 lobes. And it has a pale cytoplasm with an irregular outline containing many fine pink blue or gray blue granules. This is the neutrophils. These neutrophils actually contain two types of granules, either primary or secondary. Both of these granules are lysosomes. Lifespan of a neutrophil only 6 to 10 hours in blood. The neutrophil precursors or the site of synthesis of this neutrophil is the bone marrow. In the peripheral blood, we only see the mature neutrophil. There is no immature neutrophils in the peripheral blood because the site of the synthesis المصنع مالتنا هو موجود فقط بالbone marrow. So the neutrophil precursors actually do not normally appear in normal peripheral blood, only in the bone marrow. And the neutrophil precursors or the granulopoiesis start from a cell called the myeloblast that will mature into another cell called the promyelocyte. The myeloblast is the most immature cell, has an high NC ratio, a big nucleus in relation to the size of the cytoplasm. The chromatin or the genetic material of this cell is an immature. It does not condense. Through the process of maturation, this genetic material, this chromatin, is going to be condensed and it will be differentiated into promyelocyte. The promyelocyte will change to myelocyte, and this myelocyte will give a more granular cell called the metamyelocyte. The metamyelocyte then will differentiate into band cell and will give a mature band neutrophil, and the end result is the segmented neutrophil. This is the process of granulopoiesis. The same process occurs for neutrophil for the second component of the granulocyte, the eosinophil and the basophil. The eosinophils are similar to neutrophils actually, except the cytoplasmic granules. The cytoplasmic granules in eosinophils are coarser and more deeply red stained. And there are rarely more than 3 nuclear lobes. Actually, it's most commonly to be bilobe. They have a special role. What is the important role of eosinophil? Anybody knows? Where does the eosinophil play an important role?
Against parasitic infections?
Excellent. And what else? Another?
Against allergic infections?
Very good. The special role for eosinophil is an allergic response and defense against parasites. With the third role, that is the removal of fibrin, which is formed during the inflammatory process. The last granulocyte is the basophil, which are occasionally seen in blood. They are present in very low concentration, actually 0.1 to 1%. They have many dark cytoplasmic granules which overlie the nucleus and contain heparin and histamine. They have immunoglobulin E attachment site, and their degranulation is associated with histamine release. So they also have important role in case of allergic reaction. The entire process of granulopoiesis is under the control of what is called the myeloid growth factor. The myeloid growth factor, for example, a chemical material. These are chemical materials that regulate the process of differentiation, the process of division of different cells in order to form. These, for examples, interleukin 1, 2, 3, 5, 6, 11, the granulocyte macrophage colony stimulating factor, and the granulocyte colony stimulating factor. The process of granulopoiesis, as we said, is inside the bone marrow. And large number of band and segmented جزء من immature cells are held in the normal marrow as a reverse pool. Normally, the bone marrow contains more myeloid cells than the erythroid cell. تقريبا double up to 12 times the double. يعني المايلويد more than the erythroid. نجي other component of the phagocytes are the monocytes, which have only short time in the marrow, circulate for 20 to 40 hours, leaving the blood to enter the tissues. Then they have short time in the marrow or short time the circulation. They tend to occupy sites or tend to stay in a tissue where they mature and carry out their principal function. Their extravascular lifespan after their transformation to macrophage may be very long. إذا هم عندهم short lifespan بالbone marrow و short lifespan بالperipheral blood, بس عندهم very long lifespan بالspecific tissue where they convert to histiocyte or macrophage. They have different names according to the site they occupy. If they are in the bone marrow, they are called the histiocytes. If they are in the liver, for example, they are called Kupffer cells. But all of them, in the blood, they are called monocytes. But all of them are macrophages. The lymphocytes, they are immunologically competent cells that assess the phagocytes. هم مساعدين لعمل الفاغوسايت by activation of them. The first one is the B lymphocytes, which take the maturation process within the bone marrow and circulate in the peripheral blood until they undergo recognition of antigen. The B cell then mature into a memory B cell or plasma cell. The plasma cell, they are the cells that are responsible for the antibody production. The T lymphocyte, they develop from cells that have migrated to the thymus, where they differentiate into mature T cells. The last part of the hemopoiesis is the Megakayopoiesis, which is the process of platelet production. And the platelets actually are, the other name is the thrombocytes, are important component of the blood that are important to form the primary platelet block. And the primary platelet block is very important to stop or form the primary step in stopping the bleeding from the site of injury. The platelets in the bone marrow through the megakaryocyte fragmentation of the cytoplasm of the megakaryocyte, giving these small fragments, then that is defined as a fragment, or component of blood derived from the megakaryocyte of the bone marrow. They are non-nucleated cell, discoid in shape, small in size, 2 to 3 millimeter in greatest diameter. The process of synthesis of platelet is under control of hormone called the thrombopoietin. These are the platelets, which are non-nucleated cells with fine granules derived from the fragmentation of megakaryocyte, are wrote in black in this picture. Thank you.